This is Biology 1, Chapter 10, Part 4 of Sexual Reproduction and Genetics. In this video lecture, we will be taking a look at how to perform a dihybrid Punnett cross and also discuss probability. After Mendel concluded his experiments on monohybrid crosses, when he started to look at his results, he actually developed the law of segregation, not understanding anything about the process of cell division or meiosis, not understanding what chromosomes were, not ever using a microscope. He did and developed this law through simple visual observation, that these forms or factors that he was talking about, that one must come from one parent and the other factor must come from the other parent. So two alleles for each trait will separate during meiosis, and we call that the law of segregation. So remember, the gametes, the sperm and the egg, are haploid cells, so they only have half of the chromosomes needed to make a diploid organism. So that's why after fertilization, then you get your full set of chromosomes. Then what Mendel started to wonder is if all dominant traits were inherited together. In other words, if a garden pea plant had yellow seeds, did it always have to be tall, simply because it had yellow seeds? And did the pea plants that produced green seeds always have to be short, because those were recessive traits? And what he found through his experiments that no, the inheritance of one gene normally does not affect the inheritance of another. And we call that the law of independent assortment. There is a random distribution of alleles every time we have sexual reproduction happening. That again gives us some of that genetic variation that's so important. So, dihybrid crosses. Here we're going to take a look at two pairs of contrasting traits. So we will, again, kind of copy what Mendel did for our first example and then go into some more realistic examples. So we are going to take a homozygous round and yellow pea plant. Now remember, homozygous means the same size allele, and because round and yellow are dominant alleles, we represent those with capital letters. We'll cross that with the recessive individual. Homozygous, again, same size. In this case, it's the recessive form of the traits, wrinkled and green. Remember, we have to stick with using R's and Y's. So now your Punnett square is going to look a little more fancy than our 4-pane window. We're going to need 16 boxes in our new Punnett squares. The top and the left side will have one allele of each trait. So it will look a little weird at first, but I will show you a method to get these alleles along the top and the left side. And that guy there is Reginald Punnett. You can blame him for the dihybrid Punnett squares. So, because we need one of each trait from each parent, outside of the boxes, it is okay to have different letters together. Once we combine what we see inside the boxes, then like letters go together and capital letters come first. Now you'll notice a pattern after a few entrances here that we will have, again, if you take two purebred individuals, you will end up with a hybrid. Same situation here. Doesn't matter if it's one trait or two traits. We will have 16 heterozygous or hybrid individuals, but they will all look to have yellow seeds and they will have round seeds as well. Now, let's take a look at, we'll go back to bunny rabbits. Again, we're going to do dihybrid traits, so we're going to take a look at two types. So we will continue our rabbit fur color saying that black is dominant over brown. And fur length, we'll say short hair is dominant to long hair. So we're going to take a heterozygous short and black furred bunny and cross that with another heterozygous short and black furred bunny. Now if I'm going to use the letter S and B because those are the dominant alleles, if they are heterozygous for short is going to be capital S, lowercase s, and heterozygous for black will be capital B, lowercase b. Because the other parent is the exact same, we will use those same alleles. Now, because our box has 16 squares, and we need one of each allele along the top and the left side, we're going to use the FOIL method. You've used that in math class before. Now we're going to apply it to genetics. Okay, FOIL stands for first, outer, inner, and last, or outside, inside. I learned it outer, inner. So first, outer, inner, last. And we're going to take the first of each pair of alleles, the outer ones of each pair, the inner of each pair, and the last of each pair. So the first pairs of letters are going to be capital S, capital B. The outside of each pair of alleles is going to be the capital S and lowercase b. The inner of both pairs, or the inside letters, is going to be little s, capital B. And the last in each pair is going to be lowercase s, lowercase b. So those are the allele pairs that we can put along the outside of the boxes. And because both of our parents were the same genotype, we can put both along the top and the left side. Now remember, outside of the boxes, it's okay to have unlike letters together and for lowercase letters to come first. Inside the boxes, you combine what you see, putting like letters together and capital letters first. So you can fill out the boxes and you'll see that we are going to get a range of both genotypes, and because of that, we will have a range of phenotypes. And I will show you how to get the phenotypes from these dihybrid Punnett square problems. So that is the completed dihybrid square. Now you can see we've got a lot of different combinations, but basically there will only be 4 combinations of phenotypes that will result from this. So this is the square that you just filled out, so I will finish filling that out myself. So that's what you already have. Now this is what I do to find the phenotypes. You're going to take a look at the different combinations. Anything that has capital S something, capital B something, will have short and black fur. It doesn't matter what's in the blanks because remember, short and black are dominant traits. So I'm going to mark all of those that have capital S something, capital B something with an X, so I don't count it twice. So when you peer through the boxes, you can check off the ones that have short fur and black fur. And as we can count there, we've got 9 of those. The next combination, we will have organisms that have short fur, but it is brown. So we'll mark that with a circle. Short fur, again, is capital S something. Brown fur, because it's recessive, is always little b, little b. So I will look through my table and look for capital S something, little b, little b. I've got 3 of those short and brown furred bunnies. The next combination, I can have long fur and black fur. So I can look for little s, because that means long fur, and big B something, because that will mean that it has black fur. And I'll mark that with a square, and I have 3 of those. And by default, the last one will have both recessive characteristics of long and brown fur, but you still have to tell me how many of those there are, and we have 1 of those. So we can see this 9:3:3:1 ratio of phenotypes. We'll take a look at another example. Again, I make this stuff up. I have no idea if this is true, but we'll say that goldfish fins, their length, long, will be dominant to short, and their color, we'll say gold, will be dominant to white. So we will set up our problems. We'll take a heterozygous long and homozygous recessive white goldfish and cross that with a heterozygous long and heterozygous goldfish. Now, when I choose my letter pairs, I just go with whatever the dominant traits are, because then I can remember what the recessive ones are. So if I'm going to use the letters L and G, my first parent up top, heterozygous for long, it's capital L, lowercase l, and homozygous recessive for white is going to be little g, little g. Heterozygous for long, again, long is dominant, big L, little l. Heterozygous for gold is dominant, so we're going to have big G and little g. Now, because both of our parents are different, we are going to have to FOIL both of them. So, for my first parent, the first in each pair is going to be big L, little g. The outer of each pair will be big L, little g. The inner of each pair will be little l, little g. And the last in each pair will be little l, little g. For the other parent, again, the first in the pair, going to have big L, big G. The outside of each pair is big L, little g. The inner of each pair, little l, big G. And last, but certainly not least, little l, little g. Now, these are the allele pairs that'll go along the left and the top side. And you fill in the boxes. Like letters go together. Capital letters come first. You may have to pause it during this so you can get these filled in correctly. But remember, you can pause these at any time and rewatch any bits you like. Okay, let's go through phenotypes. Anything that is big L something, big G something, is going to have long and gold fins. So I'm going to go through and check my boxes off. I have 6 of those. My next combination, I can have short fins and gold fins. So little l, big G something. Because remember, short is recessive and gold is dominant. For the next combination of pairs, I will have big L something, little g. So we will have long and white fins. I will have 6 of those. And the last but not least, I will have both recessive characteristics of short and white fins. I will have 2 of those. We will go over how to do these in class as well. But basically what we're doing without having to actually mate things together is looking at probability. Probability is what event you're looking for, and you're dividing that between all of the possible events. So probability is the likelihood that a specific event will occur. And the Punnett squares is how we can use a paper-pencil tool to predict probabilities. This has been part 4 of Chapter 10 on sexual reproduction and genetics.